Well, thanks for the introduction, and thank you also for the invitation and for allowing me to share some of our work remotely from Santiago in Chile at the moment. I will not talk about nitrogen, nutrient, and regulation of gene expression that much today. I will focus primarily in a project that we started about 10 years ago, trying to understand the mechanisms of how plants survive in the Atacama Desert. And as you'll see from the talk, we go from, or we try to go from molecules to the ecosystem. Just to begin some motivation, you're probably all aware that human societies are facing an ecological crisis of global proportions due to the accumulation of greenhouse gases in the atmosphere, which is shifting the world's relative balance and increasing global temperatures. And this increase in global temperatures or global warming is changing climates in many parts of the world. Um, and this is, of course, a big problem for plant growth, for crop yield, and therefore for food security, biodiversity, and ecosystem integrity that I think requires urgent attention. And one of the problems that we'll see, for example, is shown in this graph, which shows the precipitation minus evaporation change on the y-axis and the latitude on the x-axis. And as you can see, particularly in the subtropic areas, which is shown in this orange or light brown color, there will be increased desertification, which in overall in the world would lead to increased global desertification from about 48 to 65% of the total land surface. And this will impact many countries, of course, countries like Chile, for example, that are located in these latitudes, but many other countries around the world will suffer from this increased aridity. And I have to say this is, or just emphasize that these are not predictions, but this is something that we're already facing. And this is one local example, which shows satellite images from a lake near Santiago from 2009 to 2019. And as you can see, it went from a lake to a complete dry place in 2019, and a location that used to be a destination for swimming and doing water activities. Now it looks like this. So this is a real problem that we are already experiencing in Chile and other countries around the world. It's the same. And I think, as I mentioned, it requires our attention, at least from the science point of view, trying to understand some of the mechanisms that plants use to face this increased aridity. It's a very important issue. And we are fortunate to be in Chile, which is, as you know, is a very long country, and as such, it has a number of climates that go from Antarctica in the very south, the Patagonia, to the Atacama Desert in the north, which is shown here. And so we have taken advantage of this natural laboratory, which is basically in our backyard, to try to understand some of these issues. So understanding how life manages to survive and thrive in some of the Earth's most extreme environments is, I think, crucial to face the future and promote social awareness also of the dire situation we face. And again, the Atacama Desert is a unique Earth system that has been actually not studied a lot. And so I think there's also great opportunities from the scientific point of view. As some of you may know, the Atacama is one of the oldest deserts and probably the driest non-polar desert on Earth. It has experienced semi-arid conditions probably for the past 150 million years. And various estimates indicate the desert inner core has been hyper-arid for roughly 10 to 15 million years ago. Thanks to a combination of unique geologic and also atmospheric conditions in this area. In addition to extreme low water availability, life here must also endure extreme daily temperature oscillations, high salinity in the soil, particularly in some parts, brutal radiation. Atacama is one of the hotspots for radiation on the planet as well. And in addition to that, which is something that brought my attention at the beginning, as a very poor soils from the nutrient point of view, primarily nitrogen, as you'll see, but also some other nutrients. which makes life difficult in addition to all the other factors that I mentioned. Despite all these very hard conditions, if we know where to look, there's surprising plant diversity. And here I show some of the species that we've been looking at over the years. And this is, of course, not looking in the oasis, but rather in the open field where they face the harshest conditions. And some interesting plants, like, for example, Solanum chilense, which is a relative of Solanum lycopersicum, the tomato. There's some lupin species as well. And of course, these, because these are relative to crops, they can be interesting to do then translation to generate new biotechnologies to improve, for example, tolerance to abiotic stresses for crops. So we have been studying a transect for over 10 years now, which is located, I will orient you here in South America, of course, in the north of Chile, in the Atacama Desert, and east of the Atacama Salt Lake. And this is an altitudinal transect that spans the limit for life. It starts at about 2,400 meters above sea level. If we go below this point, all the way to the Pacific Ocean, and we have done that transect 2 years, there is not enough water to support macroscopic life. So this is really the limit of absolute desert. So beyond this point or below this point, there is just not macroscopic life. If you go up on the western sides of the Andes Mountains, you find some life, plant life, and also animal life, all the way up to 4,600 meters above sea level. Above that, it's just too cold, and so this represents the upper limit for plant life. So between these two extremes, then we can find plant life, and we have defined 22 GPS tag experimental stations at which we have been going back every year since 2010. And one of the things that makes me happy about this project, which in the beginning we started sort of an exploratory mode, it has attracted attention from a number of colleagues around the world. And here I have some of them, and I'll show some work that we've done with Gloria Cruz at NYU, and also Dennis Stevenson at the New Botanical Garden, but also work from people in New Zealand and more recently in France for metabolomics. So I think the work that we have done has created a foundation into which we have been building more and more layers of information to try to understand molecular mechanisms of how plants survive in Atacama. So this video is just to give you a flavor for what it looks like. So we go into the field, and this is myself and a grad student. We are collecting some samples. So we take liquid nitrogen into the field. We froze the samples directly, and we take everything else that we need. If we have done some treatments, for example, then we can treat plants directly in the field. And it's not easy to work at 4,400 meters above sea level, but we've managed to do this over the years, and I'm very excited to share some of the results that we have obtained. So just to describe some of the data that we have, so we have environmental parameters. So we've been monitoring climate parameters for over 5 years now, continuous measurements every hour. And some of the data that we have collected is shown here. So, for example, temperature. So we have diurnal temperature, and we have two meteorological stations, one at about 3,000 meters and the other one at about 4,000 meters above sea level. Here you can see radiation, for example. We also collect rainfall data, soil humidity, soil water content, etc., as shown here. And perhaps I can bring your attention to this graph in particular, which is the rainfall. So in addition, as I said, so this is a very arid environment. There is some water, of course, and some precipitation, but in addition to being very little amounts of water, it falls in a very short amount of time, primarily in February, which is what's called the Bolivian summer here, which is caused by the humidity that comes from the Amazons that hits the Andes Mountain. There's condensation, and then it precipitates. And so this happens in our summer. And then from the rest of the year, there's almost no water except a few events here and there. So plants have to not only withstand prolonged periods of drought, but also take advantage of when water comes, which makes also life a little hard. We have characterized in detail the soil substrate in which the plants grow. We've done elemental analysis of the soil, and the soil is essentially the same throughout all the stations. What changes is what you can find in the soluble fraction, which is what the plants can perceive. And here I have some of these data. So, for example, you can see a pronounced pH gradient, which is alkaline soils at the bottom of the transect, at the bottom of the mountain, and acidic soils at the top of the mountain. And this is a very reproducible pH gradient. So this is data that represents 3 different years that we collected the same type of information. As you can see, this pH gradient maintained. And this pH gradient correlates well with the availability, for example, of metals, which tend to be more soluble in the acidic soils and tend to precipitate in the alkaline soils. And so this is inversely correlated to, for example, iron, magnesium, manganese, copper availability in these soils. And in the top graph here, I show macronutrients. And so nitrogen in the green here at the bottom, phosphorus, for example, you can also see a gradient, and potassium levels, which increase towards the bottom of the mountain. Salts tend to accumulate towards the bottom of the mountain due to the washing of the rains. And as you can see, so nitrogen is the primary factor in this case, which is very limiting throughout the entire transect. So this line here, 100%, represents the minimum amount that would be required for plant growth in a more normal setting, and nitrogen levels are way below that. So in combinations of this gradient of water, so increased water availability as you go up the mountain, decreased temperature, very low nitrogen availability, and high radiation throughout the transect, it really defines 3 ecosystems in the Andean Atacama. So the steppe, which is characterized by these perennial grasses at the top of the mountain, and the puna, which is dominated by shrubs and subshrubs, and is the more rich or the most diverse environment, and the prepuna, which is the most arid environment that's dominated by annual plants and some succulents, as you can see here in this picture. So one of the questions that we had at the beginning is who's there in the sense of what is the real diversity of this environment in terms of plant diversity? And this is not a simple question to answer, because in these extreme environments, several species have escape strategies. So when things get really tough, they flower, generate seeds, and they're gone. So if you don't go there at the right time, you basically miss these species. And some of them are very ephemeral. So you have some water, they germinate, growth, and flower, and die pretty quickly. And so you can have a lot of variation when we do botanical surveys, for example. And we did this to understand the diversity over 8 years, and we continue to do this, in fact. But this paper that was published a couple of years ago had this 8-year data set. But to complement botanical surveys, we did the DNA metabarcoding of the soil. We analyzed the seed bank by seedling emergent methods in the lab, and we also did macroscopic analysis of the pollen rain to try to identify the different taxa that were present in this transect. And just to summarize this, I won't go too much into detail, but I just want to say that we found 93 taxa and 78 plant species across these 3 ecosystems. So here is represented in summary the botanical survey data, where we can see the persistence, which is how often we find a species when we go in the field, look for it. And at the top, we have the annual species. Here in between, we have herbaceous perennials, and at the bottom, we have the woody perennials. And here we have all the soil proxy data. We have the barcoding DNA analysis, the pollen analysis, and the soil seed bank. And so this is important to understand what the diversity is. It's really important to have sort of a baseline of the present diversity, and which allow us, for example, to go back in time and see how this diversity has changed over the years in response to climate change, for example. And understanding that could be important to predict what's going to happen in the future. And so we did that looking at fossil samples. And these, I have to say, all this work that I'll talk today has been in collaboration with my friend and colleague Claudio Latorre, which is an ecologist that's been working in the Atacama for his entire career. And so Claudio collected these fossil rodent middens, which contains a lot of plant material, including pieces of leaf and seeds and fruits and so on. These were dated, and then we extracted a core of this sample using a facility in New Zealand in collaboration with Jamie Wood and Janet Wilmshurt at the Manaki Wanualanke Research Center. And here is Francisca Diaz, our postdoc that went there to extract these cores of these samples and extract DNA and then do the amplification in this ancient DNA laboratory, which have very clean conditions. And then once we have that, then we can use sequencing to understand or try to identify the species that were found in these samples. So using this information, we were able to reconstruct basically what the landscape in terms of the diversity has been over the past 20 to 30,000 years back, and this was also published. So I'm just summarizing the main points. And as if you go back in time, so for example, to pluvial events in the late Pleistocene and early Holocene, we see a shifting in the ecosystems where the steppe goes down the mountain and goes into the puna, and the puna also merges a little bit with the prepuna. So again, this is important once we understand how the landscape changed, how the plant species move, basically depending on the climate conditions, is important because it can help us perhaps predict what's going to happen in the future in response to climate change. But now let's focus on more on the present species. So we looked at the 32 most relevant species defined in this case based on their coverage. So how much of the landscape they cover, their persistence, or how often we find them, and also the distribution in this altitudinal range to try to get a sense for the ecological importance of these species. And this set of 32 species is shown here. This is primarily angiosperms. There is only one gymnosperm, which is Ephedra americana. So this transect is dominated by angiosperm species. They come from multiple plant families, most prominently Poaceae and Asteraceae, but there's others, as I mentioned before, Solanaceae and also Fabaceae. They have different types of photosynthesis, life forms, and about a third of them are endemic species, which, as you may imagine, there's very little known about them. And in fact, in most of these species, besides their botanical names, there's very little information, molecular information about that. So most of what I'm going to talk about is published in this paper from last year. So if you want to know more details, you can go to that publication. And here again is represented the altitudinal transect from 2,400 meters, at the bottom of the mountain to 4,500 meters, which is the highest station. And in this blue case, in these blue squares now is represented the persistence for all these species that we collected. And so we collected these 32 most relevant species as we define them, and we sequenced the transcriptome using Illumina technology. And here I show pictures of all of them so that you have a sense of the type of species that we have characterized. Again, some lupin species, 2 lupin species, this Solanum quilense, which I think is also very interesting, and several grasses, which of course are related to some of the grasses that are cultivated. I'm not going to go into too much details about this, just to say it took us quite a bit of time to do the novel transcriptome assembly, to set up the pipeline to do this analysis. So all the raw reads, of course, were processed for quality and then assemble. And then we use these assemblies to perform protein predictions. And once we have this set of non-redundant proteins, we use BLAST and other tools to annotate them and try to understand some of the functions that were represented in these transcriptome data sets. Just to give you a sense for this the scope of this project in this environment. So this is a picture that I took in one of our field works, and basically because of the criteria that we use, which is coverage, persistence, and distribution. So basically, we have sequence information for all the species that you can see in this picture, because the remaining of the species are very, they're not very abundant, or they come and go every, I don't know, 3 4 years, depending on the climate conditions. So in a sense, I think it gives a very complete information about the entire ecosystem with regards to the plants, at least. So the question then that we wanted to ask with these data sets is, what are the features of these Atacama species that allow them to survive? And one of the first things we did, and this is in collaboration with a colleague from Universidad de Chile, Mauricio González, is to look at some of the partners that the Atacama plants have. And as you know, the plants live in close association with microorganisms, with bacteria. And with Mauricio, what we did was to analyze the microbiome associated with the Atacama plants to try to see whether there were specific functions that may be relevant for survival. So what's shown here again are the different sample species, the plant species, different taxonomic families here in the same colors, and the belt, which refers to which environment they were collected in. So Prepuna, Puna, Stepa, as I showed, the three vegetational belts found across these transects. So what was done here, and I will just again summarize the main results. So we analyzed the bare soil. So we collected bare soil. This is soil close to the species but that didn't have plants in the proximity, and 16S ribosomal RNA was sequenced, and the OTUs were classified. And as you can see here, there is a prominent presence of proteobacteria, and also actinobacteria and acidobacteria. And then you can contrast the distribution of bacterial taxa to the microbiome that is associated to the plant. So these are samples that were obtained from soil close to the root system of all these species. And there is a significant change in some of the taxa, and here just shown for these major bacterial phyla. So proteobacteria, for example, increase in the root zone soil. Bacteroidetes also increase, and there is a decrease in actinobacteria, for example. So to try to understand the implications in terms of the functions that may be related to this selection or enrichment of specific taxa near the plants, we perform functional prediction using the sequence information. And so we could see, for example, so these colors represent the enrichment in the root zone with regards to the bare soil. So the purplish, or the more purple the color is, the more enriched this function is in the root zone. The greener is the less enriched, or is more enriched in the bare soil, if you wish. And as you can see, so there's a number of functions that are enriched for some of the species in the root zone. In particular, that calls our attention is, for example, this nitrogen fixation. And we did as a marker to try to get a validation of this functional prediction. And here we have the information for the NIF-H marker gene, which is a marker for the presence of the nitrogenase, which is an important enzyme for nitrogen fixation in bacteria. And as you can see, there's many species of plants that appears to have increased diversity of NIF-H containing taxa near the roots. And this, of course, even though we don't have yet proof that there is nitrogen fixation in the soil, it makes sense that because these systems are so nitrogen depleted, that plants would recruit nitrogen fixing bacteria to help them cope with that aspect, at least. Now, to try to understand from the plant point of view what was unique about these species, and we had a big data set. So these were close to 890,000 total protein coding genes that we identified, 486,000 plant protein coding genes approximately, and more than 300,000 new protein coding genes. So this is a big data set. So how can we narrow this big data set to try to understand functions that may contribute to adaptation in Atacama. And so the strategy here was to compare this transcriptome information of the Atacama species to the information or the sequence information of sister species. And these are phylogenetically related species, but that don't live in Atacama. So presumably they're not adapted to cope with these very stressful conditions, right? Because they don't live there, they live in more mesic environments in many cases. And we also threw in the mix 6 model species just to bring the annotation information, such as Arabidopsis, maize, and soybean. So in order to identify these important genes that can contribute to biological processes for adaptation, we collaborated with a group of Claire Carusi at the New York University, which they have developed this big plant pipeline to define orthologs, reconstruct relationships that then lets you or allows you to narrow down to candidate genes that may explain some of the adaptation of these species. So we took the 70 species that I mentioned, so the 32 Atacama species plus 32 sister species and the 6 reference genomes for a complete data set of 1,666 plus input sequences, a big data set. This was a huge task, but I'll summarize it in a couple of minutes. So the first step, so in this first row, basically what it's done is to identify orthologs. And to do this, so there's a gene clustering step using BLAST, MCL, then alignments using MAFT, and then these trees, gene family trees are conducted for every gene family in the data set, from which then one could define the orthologs. And once the orthologs are defined, they can be used to create a phylogenetic matrix, which is basically all the orthologs for all the species. So in each row of this matrix, you have a species. So for example, here, species A, species B, species C. And then in all the columns, you have basically the protein sequences, right? And so this is a huge phylogenetic matrix that has all the species and all the protein sequences for which we can identify orthologs. And this is then used to build a total evidence tree from which one then can analyze characters that are important for the divergence of the species. So for example, using methods such as DNDS or positive selection to try to identify what are the protein sequences that are under positive selection and where these protein sequences play a role in what biological processes. And in this way, we can identify molecular mechanisms that may contribute to adaptation. So here are the numbers. So we build gene family trees for over 32,000 families. As again, this was a huge computational job that the people at NYU solve. These allow us to identify almost 16,000 orthologs, and with this we built this phylogenetic matrix that had 8,599,000 plus amino acids in length. So these are the columns of this matrix, right, and the 7 species on the rows. And with this, we build, as I said, the total evidence trees from which then we can go on to analyze specific character changes that may explain the structure of this tree. So this is a tree. It probably doesn't look very exciting because it looks like the NCBI tree, but this is good, right? Because it was built using the sequence information. And so what interests us here is, for example, branches like this, right, where you have Atacama species like Jaraba frigida and Nocella nardoides, which are 2 grasses that grow at the top of the transect, and compare these grasses to Stipa grandis, which is a related species, but that doesn't live in Atacama, of course. And so by understanding what are the changes that distinguish these species, we hope to have a sense of what's important for adaptation in Atacama. So again, so this was a big computational job, 1.7 million proteins approx, and took about 1.5 months of runtime once the pipeline was established just to run the tree. And this work was done by Kranti Barala, a great collaborator, which is now an assistant professor at Purdue University. So next, so once we have this tree built, Gil Eschel perform positive selection analysis to try to uncover important genes and processes, again, for survival of these plants in the Atacama Desert. And he used two main methods. So first, a tree-dependent DNDS modeling approach using CodeML, branch site model. And here we use species-independent adaptation and ancestral adaptation to try to identify positive selected genes. And then the second method was a tree-independent DNDS calculation using SNP Genie, where basically we compare groups. So here the different groups were built. So for example, we compare the Atacama species versus the drought-adapted sister species, or the Atacama species versus the mesic sister species. Or we use drought-adapted sister versus mesic sister species, just to try to parse out the drought adaptation, or all Atacama versus sister species to try to parse out the nitrogen adaptation, for example. So all these different combinations give us different sets of protein sequences at their positive selection. And overall, we found 265 gene families that were under positive selection in the Atacama plants species. And this graph summarizes that. So here at the top, I have the species again. In red, I show the Atacama species. In black, we have the sister species, and in blue are the model species. Again, these colors represent the taxonomic plant family. So in yellow, the Asteraceae, in green, the Poaceae, which are the main groups. And here in the middle of this graph, in gray, are shown cases where you have genes, families under positive selection in each of the species. So as you can see, there's a significant group of genes that are under positive selection in the grasses. Then there is another set here that is under positive selection in the Steraces. So this represents about 36%. These are plant family specific genes that are under positive selection. The rest, 2/3 basically, are under positive selection in at least two different gene families. And this is important because these plant species come from different evolutionary trajectories, is evidence for converging adaptation in many cases. And in some of them, as you can see here at the bottom, so these gene families are found under positive selection in most of the species in the transect. And here at the right side, I have different methods that were used that provide evidence for the positive selection. And in the last column, I think this is also interesting because it shows the relative level of expression of these gene families. And the browner the color, it represents the higher the expression levels of these corresponding genes. And so it indicates these genes are not only under positive selection at the protein sequence level, but they are also, in many cases, very highly expressed, which probably tells you that on one hand, that they're probably doing a job in the field, but also that they may also be under regulatory selection of some kind, which of course we cannot parse out here, but it's suggested by the data. Now, to understand some of the biochemical functions that may be involved in the survival of these plants in the Atacama, we first look at the PFAM domains. So these are protein domains that are annotated in the PFAM database that gives you a sense for what this protein sequence may encode, whether it's a kinase domain or a chaperone domain or transcription factor, for example. I'm sorry. So the first thing, in many cases, 67% of the protein sequences that we had an annotated domain. So that was the first thing. And in many cases, 64% of these domains have the positive selection site. So again, this does improve, but it's an interesting correlation that may indicate that these positive selection events, so these changes in the protein sequence that is selected in Atacama, may be related to functional change because it occurs within defined functional domains of proteins. And some of these protein domains, Some of these protein domains are indicated here. So some of the most prominent are the protein kinase domains, DNA-J domains. Some of them we can identify or associate with stress functions, and some have, of course, more general functions. But this is a first approach to try to understand what these proteins may be doing. Some of them are overrepresented PFAM domains with regards to, how often you are expected to find these domains in a set of proteins of the same size as we have in our data set, like SNF7, for example, or zinc fingers, and so on, or DNA-J domain as shown here with the blue star. Now, second level of annotation that we can use to try to understand the processes that are involved in adaptation is the gene ontology terms annotation. So we were able to annotate 180 of the positively selected genes in our data set, and the processes where these genes are appear to participate based on the genotype annotation are summarized in these slides. So here at the center of these clusters, we have the main processes, so response to stimuli, biological regulation, developmental processes, metabolic processes, cell component organization, and localization. The color of the squares represent the number of positively selected genes that are associated to those functions. So as you can see, for example, cellular nitrogen compound biosynthetic process is a very prominent process. And again, it makes sense if you think about the environment where these plants live, which is depleted for nitrogen, it has very little nitrogen. Also, you can see here response to light stimulus, response to reactive oxygen species, which of course are related to the high radiation that these plants encounter, and so on. So again, so these biological processes appear to be related to the environmental stresses these plants face in this environment. Some of these are overrepresented, like, for example, again, cellular nitrogen compound biosynthetic process. And finally, to try to understand or to validate the functions of these genes in terms of their capacity for adaptation for these plant species, we use orthologs from our Arabidopsis for the positively selected genes from Atacama species, and try to see what kind of information we have for these genes in this model species Atacama. And we found many genes associated to, for example, nutrient use efficiency, both nitrogen and also phosphate. We find genes associated to root development, such as enhancing root hair growth, for example, or optimizing water capture. Also genes involved in biotic interactions. And in above ground processes, we find genes involved in modulating flowering time, which of course is very important if you think about what I talked at the beginning. If you have water, for example, that very unexpected time, and you have to complete your life cycle very quickly, I think it's essential to modulate all the pathways related to flowering time. We also find genes involved in photosynthesis, both protecting the photosynthetic apparatus as well as improving photosynthetic performance. And finally, also many genes involved in coping with high radiation. So in many cases, which are all these cases that are marked with this dotted blue line on the outside of these gray ovals, there is genetic evidence that these genes do participate in these stress tolerance phenotypes related to all these processes that I mentioned. In many cases, of course, also which are marked with this blue bar with the red circle, we have positive selection within the PFAM domain, which again suggests there is a functional change in these proteins that may contribute to adaptation. And also in several instances that are marked with these stars, these genes are very highly expressed in at least 2 Atacama species. So again, indicating this, there may not only be a selection at the protein sequence level, but there is also these genes are playing a role because they are highly expressed, and perhaps they also are under selection at the regulatory level. So overall, we could validate more than 30% of the positively selected genes using the information in Arabidopsis, and the remaining 70%, or I think it's really a genetic gold mine that one could use to try to understand new processes that may contribute to adaptation or new aspects of known processes. So I hope I've shown you how we can go from big data sets at the molecular level using integrative bioinformatics and, in this case, phylogenetic models to generate primarily a number of hypotheses that we hope may contribute to face some of the big challenges that we have with regards to climate change, as I mentioned at the beginning. And I think this iteration between big data sets, models, and hypotheses building and testing is really what's driving science forward, at least in our experience. We have identified a number of new genes, which I think may be important for survival in Atacama, and of course, the work we're doing, and which is probably going to take us a number of years, is to follow up on some of these interesting genes and of course validate their function. So with that, I would like to just acknowledge some of the people that was involved in this project. As I said, this is a very long project. There's been a number of publications coming associated to it, and the work that I talked today is primarily the work of Gil Eshel and Viviana Arauz, at least at the later stages. But for example, Daniela Soto, Carol Moraga, and Alejandro Montesinos and Tomás Moyano also were instrumental in the bioinformatic analysis. Soledad did also a lot of the work in the soil analysis, and many other people from my group, which is shown here in the last picture, previous to the pandemic, contributing in different ways. This project was funded initially actually by Howard Hughes, which was great because it allowed us to explore different things. Later was funded by funding from Chile, the Center for Genome Regulation, and the collaboration with Gloria was funded by the Department of Energy, where we were also able to build a similar data set to the Atacama plant species, but from the California desert. And this work is coming probably in the coming year. Here I have some pictures for the many people that participate. So thanks to many of the people that participate in the field work. So always Claudio here, my colleague and friend, but different students, technicians, and postdocs, and some colleagues that visited from different places. And every year the crowd was slightly different, always Claudio and me, but the rest may change. Also, I have to acknowledge the Evonet team. So the people at NYU, as I mentioned, was instrumental to do the phylogenomic analysis of Gloria's group, Kranti Varala and Gil Eshel. Also the work with Dennis Stevenson, which has been related to identifying sister species and also finding the California relatives. And Rob Desal, which also was very, very important for all the phylogenetic work that we did. As I mentioned already, Claudio has been, without him, this project would have not been possible. And Mauricio Gonzalez, which is a colleague from Universidad de Chile, is also very important for all the microbiology work that I briefly described in this presentation. So with that, I would be happy to take questions if there are, and I leave you with this quote from a Chilean poet related to the Atacama Desert. Thank you very much.
Okay, thank you, Rodrigo, for your great talk. So have time for questions.
Well, in the literature, have you found that in other deserts, or in other arid areas, it has been reported that there are that the plants use similar strategies that those that you found in the Atacama Desert. Well, that's the first question, and the second question, it's the have you found any relationship? Because well, usually when you have in the rhizosphere communities of bacteria, you interacting with the roots of plants. They're also usually interacting with some kinds of fungus because the filaments of this fungus are thinner than the roots of the plants, and they're able to get water from and nutrients and transport that water and nutrients from places that the roots are unable to uh, arrive. No, so in your study, did you found the communities of bacteria that could be related with some kinds of fungus? Well, that two questions.
Okay. Thank you very much. Yeah, those are very good questions, both of them. So with regards to the first one, so there are some mechanisms that are that will be shared by other plant species in general. As I mentioned, when I showed this graph with this gray square, so in some cases we see conservation across all the plant species, and these are also shared by other species, even things like Arabidopsis. So there's definitely some mechanisms that are common to different plant species, not only in deserts, but also regular plants to cope with drought. But there's also cases that are specific to Atacama, and there is also cases that are probably species specific. So we have the different levels. In this case, I chose to focus on the more common or the convergent solutions, but there's definitely also specific solutions that may be also very interesting to study in the future. And for the second question, we haven't looked at the fungi. I have a colleague in another Chilean university, which is actually doing that kind of work. So she is isolating fungi that are associated to the roots and then testing them in laboratory conditions to see what effect do they have. And she's found some interesting strains that can promote growth and also protect from both abiotic and biotic stresses as well. So this is also a very interesting, I think, area of development. So try to understand what these microorganisms do, both bacteria, but also fungi.
Thank you.
Hi. Um, in your experience, the adaptations mainly belong to genes that encode for conserved metabolism, so for genes that are part of the core genome, or or the main adaptations are more in the specialized metabolism and are recent gene acquisition or something like that.
Yeah, very interesting. So we have a couple publications already out, one in Neophytologist recently, where we looked at the metabolism specifically. So we use, this is work done in collaboration with our colleagues at Bordeaux University, with Pierre and Yves Gibbons, in Yves Gibbons' group. And it's a very interesting story there because so we have some metabolites that appear to be part of the central metabolism, and also some metabolites that appear to be more specialized or secondary metabolism. So I would say the answer to your question is both. But what's interesting is that there appears to be a core set of metabolites that are important for adaptation in many of these species. So in fact, we have identified about 30 of them that appear to be relevant for all these species, which also suggests that this will be also important for adaptation or for tolerance to, I would say, not adaptation to, for tolerance to abiotic stresses in other species such as crop. And this is a little bit different from the maybe the current understanding of how this goes, where there's more specialized solutions in many cases to the environment. I don't know. Did that make sense? So
Yeah.
basically, we have primary metabolism, secondary metabolism, but there seems to be a convergent solution to respond to these environmental stresses.
Yeah, and some proportions that you have in mind, or no?
No, I don't really remember the proportions. But
Okay,
if you send me an email, I can tell you later.
thank you.
Hi. I was just wondering if within the genes that you found under positive selection, did you find any gene associated with salinity, like an adaptation to high tolerance to salinity? I didn't get that, sorry. And the other question is, in the genes that you found that had this signal of positive selection, how different are these enzymes from Arabidopsis? Like, have you made a tree to see how divergent they are? How can we say that they share the same function? That's what I was wondering.
Right. Yeah, very good question. So there's a number of proteins that could participate in salt tolerance. So transporters, for example, that we found that may be important for that. Although, as I mentioned, we haven't yet proven the function in these genes. This is something that is underway. So this is basically done at the sequence level analysis and then in comparison to what's been done in other species, such as Arabidopsis. So in this case, related to your second question, it depends on the gene family. So some gene families are quite conserved. So even though of course, we cannot say it's playing exactly the same function, given that the protein sequence is very close, one can presume that it's related. It's performing a related biological function or participating in a similar biological processes in these Atacama species. But again, so this is something that it's for future studies that will allow us to really validate these mechanisms. So in fact, this is something we're doing at the moment. So trying to introduce some of these genes into models such as Rhabdopsis or maize to validate their function and really perhaps use them as biotechnology tools for crop improvement.
Thank you.
Hi, thank you for the presentation. So I have two quick questions. The first one regarding the life cycle of the plants you analyzed. So did you stratify your data according to the life cycle of the plants? Some of them complete their life cycle in 1 year or less, or in months, while in the Atacama Desert you also have some perennials such as cacti, which take like a whole lifetime in there. And the second one is when working with transcriptomes, you are dealing mostly with coding sequences, but are there any plans to look at the wiring of the gene regulatory networks, maybe exploring long non-coding RNAs or some other RNAs that could modulate the expression of these coding sequences?
Yeah, also very good question. So yeah, as for the first question, so we did the analysis and comparing the different life forms, so whether they're perennials or woody perennial, herbaceous perennials, or annuals. And although there are some differences, so some positively selected genes are found in specific types, the core is shared by all these species. So it appears to be independent of the growth habits or the life form of the plant. We also did, just to complement, the same type of analysis regarding to where the species grew, whether it was at the most arid environment at the bottom of the transect, or it was at the coldest environment at the top of the transect. And again, so the majority of these positively selected genes we found, because of the way we did the analysis, of course, were shared among all these species. Because again, so we were trying to find these conversion processes, so gene that were under positive selection in multiple gene families. So one could do a more, directed analysis to try to dissect better, for example, differences between the perennials and the annuals. Yeah, but that's not the way we chose to do this analysis in particular. And what was the second question? I lost track now.
Yeah. So the second one was more about sometimes the coding sequences did not evolve that fast to allow
Oh, right.
for
Yeah, right.
adaptations.
Yeah.
Maybe are there any plans at looking for non-coding RNAs that might be rewiring these gene regulatory networks?
Yeah. Okay, yeah, sure. That's also a very good question. We haven't done that yet exhaustively. So we did some collaboration with Marie-France Sagot to look at the microRNAs, for example. But we haven't followed that much on that line of work. Of course, that would be very interesting to complement the focus on the protein coding sequences. But there is so much to do. So if any of you, of course, is interested, I'll be happy to share data to look at that aspect.